Microbial metabolisms involve how tiny organisms use chemical processes for energy and sustenance. Understanding their metabolic pathways is like learning the steps in a recipe, and it's crucial for comprehending how they operate. Analyzing the diversity in microbial metabolisms explores the various ways microbes use to survive in different environments. This knowledge is essential because it helps us understand the power of microbes in biotechnology, where they play vital roles in activities like environmental cleanup, biofuel production, and drug development, relying on their metabolic skills for these applications. Before we go into detailed discussion, let's take a look on some important definitions and principles on metabolisms. Metabolism in living organisms involves all the chemical reactions, which are made possible with the help of special proteins called enzymes. These reactions can be divided into two types: catabolic reactions, which break down molecules and release energy, and anabolic reactions, which build molecules and require energy. Enzymes are vital in this process, as they are protein molecules that speed up these chemical reactions by reducing the energy needed for them to happen. Enzymes are highly specific, meaning they work on particular molecules. Metabolic pathways are like sequences of chemical reactions that occur one after the other to achieve specific outcomes in the cell. Biochemical processes involve several important functions, including first, the transfer of functional groups, where one molecule hands over a specific group to another; second, the creation or removal of carbon-carbon double bonds, which includes processes like hydrogenation, adding hydrogen atoms; third, isomerization, rearranging atoms in a molecule; then we have ligation and hydrolysis, referring to the joining and breaking of molecules; and finally, oxidation-reduction reactions, where molecules either lose or gain electrons. These processes are fundamental in the intricate world of biochemistry, governing how living organisms transform and utilize molecules for various essential functions. Metabolism is crucial for various reasons. Firstly, it's responsible for energy production by extracting energy from nutrients, which is essential for all cellular activities. This energy is often stored in molecules like ATP, acting as the cell's power source. Secondly, metabolism is vital for growth and repair, with anabolic pathways synthesizing complex molecules like proteins and DNA needed for cell development and maintenance. Additionally, metabolism plays a role in waste elimination, detoxifying and removing harmful substances from living organisms. Furthermore, it contributes to the regulation of homeostasis by maintaining factors like temperature, pH, and nutrient levels within a stable range. Lastly, metabolism enables adaptation to environmental changes, allowing organisms to switch between different energy sources based on nutrient availability, which is crucial for survival and thriving in various conditions. This image here shows how metabolism occurs in a bacteria cell. Nutrients were absorbed from its surrounding, broken down or catabolized inside the cell, providing energy for synthesis of new molecules including cellular structures and new cells. This synthesis pathway is known as anabolism. Now that we have understood the concept of metabolisms, let's explore how this concept can be used in the classification of microorganisms. Microorganisms are classified based on their metabolic processes, which involve how they get energy and carbon. Some microbes use light for energy are known as phototrophs, while others rely on chemical compounds or chemotrophs. In terms of carbon sources, autotrophs use carbon dioxide, while heterotrophs need organic or inorganic compounds. Additionally, microorganisms can also be classified based on the type of electron donor they use, with some depending on organic sources and others on inorganic sources. By looking at these metabolic characteristics, scientists can sort microorganisms into different groups, helping us understand their roles in the environment and their potential applications in various fields. Try to answer this question based on the classification mentioned earlier. How are microorganisms that derive their carbon from chemical compounds and their energy from light sources classified, and what are some examples of these microbes? Therefore, all organisms can be classified based on their metabolic properties. This figure here shows the flowchart on the classification of bacteria, based on energy sources, carbon sources, and final electron acceptor. Note the presence of bacteria relevant to environmental processes. How are they being classified? All biochemical reactions occur in living organisms are catalyzed by protein molecules known as enzyme. The next discussion will explore on enzyme, its structure and functions, how enzymes are being classified and the factors affecting enzyme activity, including the effects of inhibitors. Enzymes are biological molecules that act as catalysts, facilitating and speeding up chemical reactions in living organisms. Enzyme reduces the time taken to complete a reaction by lowering the activation energy. They are typically proteins with specific three-dimensional structures that include an active site, a region where substrates bind and undergo chemical transformations. During this process, the enzyme itself remains unchanged. Enzymes are highly specific, meaning they catalyze specific reactions, and their function is crucial for maintaining life processes. The activity of an enzyme can be measured by measuring the change in concentration of substrate and product. Enzymes have a complex structure crucial for their function. The reaction occurs at the catalytic site, a specific region within the enzyme where substrates bind and undergo chemical transformations. Some enzymes require cofactors, which can be metal ions like zinc or magnesium, vitamins, or other non-protein molecules, to assist in catalysis. Coenzymes, a type of cofactor, are organic molecules that often shuttle chemical groups between enzymes, such as NAD+ and FAD in energy metabolism. In many cases, enzymes are inactive without their necessary cofactors, as these molecules are essential for the enzyme to adopt its proper conformation and facilitate efficient chemical reactions. Cofactors and coenzymes play a vital role in ensuring the enzymatic reactions proceed with precision and efficiency. Enzymes are categorized into different classes based on their specific functions and the types of reactions they catalyze. Oxidoreductases are involved in redox reactions, transferring electrons between molecules, with alcohol dehydrogenase as an example. Transferases facilitate the transfer of functional groups, exemplified by phosphofructokinase in glycolysis. Hydrolases break chemical bonds through the addition of water, including proteases that cleave proteins. Lyases catalyze reactions involving the addition or removal of functional groups without using water, with fumarase as an illustration. Isomerases rearrange atoms within molecules, such as glucose isomerase, converting glucose into fructose. Ligases, represented by DNA ligase, are responsible for joining molecules, typically requiring energy from ATP, playing a critical role in DNA repair and genetic processes. Now let's identify the things that can affect the efficiency of enzyme activity. The first factor affecting enzyme activity is temperature. The rate of an enzyme-catalyzed reaction generally increases as the temperature rises because higher temperatures provide more kinetic energy to reactant molecules, allowing them to collide more frequently and with greater energy, which can enhance the reaction rate. However, there is an optimal temperature for each enzyme, known as the optimum temperature, at which it exhibits its highest activity. Beyond this point, a continual increase in temperature can lead to a sharp decrease in enzyme activity. This decline occurs because the excessive heat disrupts the enzyme's three-dimensional structure, particularly the active site, causing it to change shape. When the enzyme becomes denatured, it loses its catalytic function, rendering it incapable of facilitating the reaction. In essence, the enzyme's temperature sensitivity highlights the delicate balance between enhanced activity and thermal damage, emphasizing the importance of maintaining suitable temperature conditions for enzyme-driven reactions. Next, the pH. Changes in pH can indeed affect the shape of an enzyme's active site. Each enzyme has an optimal pH at which it functions most efficiently, and this optimal pH value varies depending on the enzyme's natural environment or where it typically operates. For instance, enzymes in the small intestine, where the environment is slightly alkaline, have an optimum pH of about 7.5. In contrast, stomach enzymes, which operate in the highly acidic environment of the stomach, have an optimal pH of about 2. This pH dependence is crucial because it ensures that enzymes work optimally in their respective body compartments, where the pH conditions are tailored to their specific functions, underscoring the importance of pH regulation in enzyme activity and overall digestion. The concentration of substrate also plays an important role in defining the enzyme activity. As the concentration of the substrate increases, the rate of enzyme activity indeed rises due to more frequent collisions between enzymes and substrates. However, this increase in rate is not indefinite. Enzymes eventually become saturated, meaning that all available enzyme active sites are occupied by substrates at any given time, and further increases in substrate concentration do not lead to a corresponding increase in the reaction rate. This phenomenon is often described as the enzyme reaching its maximum reaction velocity or saturation point. In such a state, the enzyme is working at its maximum capacity, and the rate of the reaction levels off, emphasizing that there's a limit to how fast an enzyme can catalyze a reaction, even if an excess of substrate is present. Now, let's take a look at another factor which can affect the enzyme activity, the inhibitor molecules. An enzyme inhibitor is a molecule that blocks or hinders an enzyme's activity. There are two main types: competitive inhibitors, which directly compete with the substrate by binding to the enzyme's active site, and non-competitive inhibitors, which attach to a different site on the enzyme, causing it to change shape and reduce its ability to bind the substrate. In addition to this, inhibitors can be reversible, meaning they can be removed, allowing the enzyme to regain its activity, or irreversible, forming a strong bond with the enzyme and permanently inhibiting it. Inhibitors are essential for regulating enzyme activity and have various applications in biochemistry and medicine. This is some examples of enzymes which are widely being used in industrial processes. Enzymes are extensively utilized in industry as biocatalysts for a wide range of processes. They enable the efficient production of numerous products, including food and beverages, textiles, detergents, and biofuels, by facilitating specific chemical reactions. Enzymes offer environmental benefits, such as reduced energy consumption and waste production, making them essential for sustainable and cost-effective industrial processes. In the food industry, they aid in enhancing flavors, prolonging shelf life, and converting raw materials into valuable products. Their versatility and eco-friendly nature make enzymes valuable tools for optimizing various industrial processes and reducing the environmental footprint of manufacturing. That is all for this time. Until we meet again on the next topic. Thank you and have a great day.